Hello children, good morning. So yesterday we have discussed about double helix structure of DNA, and we have also discussed about the different silent features of double helix structure of DNA, right? So Francis Crick also proposed the central dogma in molecular biology, which states that the genetic information actually flows from DNA to RNA and then from RNA to proteins, right? So the process of making a DNA copying from itself, that process is known as replication. It is clearly shown in the picture. The process of formation of mRNA from DNA is known as transcription, and the protein synthesis from RNA is known as translation. So this is known as central dogma. Children, in some of the viruses, the flow of information is in reverse direction. That is from RNA to DNA, right? So that process, that way is known as central dogma, right? So in central dogma, we will discuss about DNA, replication, we will also study about transcription, we will also see about the translation, right? Now children, with that we got an idea about the structure of DNA, right? Now we are aware about the structure of DNA. Children, now the question is, where is the DNA located? Now, as I already discussed in very start of this lesson, that DNA is located inside the nucleus, right? If we try to calculate the length of DNA, it is found that it is far greater than the dimension of nucleus. That is very strange, right? Like, length of the DNA is found to be far greater than the dimension of typical nucleus, but still it is located in the nucleus. Because it is something like that. Let us suppose you have a house to stay and your size is bigger than the house. So in that case, we are not able to stay in that house as shown in this picture, because house capable to accommodate you, but the house is very small, so it is not in the condition to accommodate you, right? It is something like that. Children, when you were kid, you used to play with baby dolls, right? Dolls से आप खेलते थे. So with doll you have to with their small houses, और dolls के साथ उनके छोटे छोटे घर भी होते हैं, right? Doll house जिसे कहते हैं. So the small house were good enough for dolls, right? But you cannot stay in that house. So in this picture it is clearly seen that the doll is able to stay in this house, but you, little girl, you are not able to stay in that house because that was very small for you. Now in the very similar way, if you talk about the packaging of DNA, right? Now if the size of DNA is going to greater than the size of nucleus, how will the DNA fit it in that nucleus? Now before getting to that discussion, first let us try to look at the calculating the length of DNA, right? So let us try to calculate the length of DNA. Now just now we saw that how DNA is actually organized. It is organized in the form of base pair, right? जो DNA होता है वो organized होता है in the form of base pairs. So by looking at the number of base pairs, we can actually estimate the length of DNA. So greater is the number of base pair would be the length of DNA. So that is the common logic. Now the total number of base pair which is seen in any typical mammalian cell, right? अगर हम एक typical mammalian cell में base pairs की बात करते हैं, so it is around 6.6 into 10 to power 9, right? So total number of base pair in any mammalian cell, right? Now we know that the distance between two base pair, like the distance between two base pair, yesterday we have seen it is 0.34 nanometer. इसको हम कैसे लिख सकते हैं? इसको हम लिख सकते हैं 0.34 इंटू 10⁻⁹ मीटर. If I have calculate length of DNA, so we know the distance between two base pair, right? The distance between two base pair is 0.34 into 10⁻⁹ meter. So its total length of base pair is 20 in a day DNA, right? If we suppose that 20 base pair है किसी भी DNA की length, so length of DNA is equals to DNA की length, फॉर्मूला अगर हम देखें, DNA की length is equals to distance between two base pair into total base pairs. अगर मैंने माना कि DNA में 20 base pairs हैं, तो हम कैसे निकालेंगे DNA की length? हम DNA की length निकालेंगे, नंबर ऑफ बेस पेयर कितना है? नंबर ऑफ बेस पेयर इज 20 और दो बेस पेयर के बीच का डिस्टेंस 0.34 इंटू 10⁻⁹. तो डीएनए की लेंथ कितनी आ गई? अगर आप इसको कैलकुलेट करोगे तो तकरीबन ये आपका आएगा 4 इंटू 10⁻⁶, मतलब कि तकरीबन 2.2 मीटर्स. So this is the length of DNA of typical mammalian cell. Now what is the size of a nucleus? Nucleus का size कितना होता है? It is common, we all know that the size of the nucleus is of order of 10⁻⁶ meters, right? So there is a huge difference, right? DNA कितना है? 2.2 मीटर और न्यूक्लियस का साइज कितना है? 10⁻⁶ मीटर्स. सो इट शोज दैट डीएनए लेंथ इज वेरी लार्ज, नॉट ओनली लार्ज बट वेरी लार्ज. We compare to this the size of nucleus, but we say that DNA is located inside the nucleus, so that is so strange. So how is the DNA packed in the nucleus? But फिर भी we are very sure that it is located in the nucleus, right? So that is why understanding DNA helix package is become very very important, right? तो इसलिए DNA पैकेजिंग जो है वो पढ़ना बहुत ही ज़्यादा इम्पोर्टेंट हो जाता है. सो नाउ वी विल सी हाउ एग्जैक्टली इट इज़ पैकेज इनसाइड द न्यूक्लियस, राइट? सो लेट अस फर्स्ट टॉक अबाउट प्रोकैरियोट्स. Now why we are taking separately in prokaryotes and eukaryotes? In both of them, infrastructure inside the cell is very different. In eukaryotes, we see each organelle separate, but in prokaryote, we don't have all these things. So let us first discuss about prokaryotes. That where do we have DNA? So in the case of prokaryotes, DNA is organized with loops held by protein. So it is not that DNA is scattered throughout the cytoplasm of prokaryotic cell, because in prokaryotes membrane bound cell organelles are absent. That means DNA is scattered in the cell. No, that is not true. What happens in that? DNA is concentrated in one region and DNA is get organized into loops. Okay, DNA का कोई मेम्ब्रेन बाउंड सेल ऑर्गेनल नहीं होता. इसका मतलब ये नहीं है कि डीएनए साइटोप्लाज्म में जहाँ तहाँ स्कैटर्ड फॉर्म में प्रेजेंट है. डीएनए इज कंसंट्रेटेड इन वन रीजन ओनली. डीएनए इज गेट ऑर्गेनाइज्ड इनटू द लूप्स. So here it can see thin thread like structures in the form of loops. Okay, now DNA is being negatively charged. So why DNA is negatively charged? DNA जो होता है वो negatively charged होता है. क्यों? Because of the presence of negatively charged phosphate group. क्योंकि DNA में negatively charged phosphate group होता है, इसलिए DNA भी negatively charged होता है. So everywhere you see the phosphate group in the polynucleotide chain, they are negatively charged. So DNA is also negatively charged. Whereas talking about the protein, several protein which is present inside, they are positively charged. So we all know that opposite charge attract each other. So what happens? DNA and protein get attracted to each other and DNA loop around the protein, and with their help they organized in loops. So the region where DNA is present is termed as nucleosome, or simply it termed as nucleoid, which is the primitive type of nucleus. And this region where most of the genetic material or most of the DNA is concentrated, it is known as nucleoid, right? So this is the name given to the DNA rich region inside the prokaryotic cells. So this is how DNA packed inside the nucleus. So here also size of nucleus is smaller than the size of DNA. But how DNA is able to accommodate it? Because it is present in the form of coiled structure. So it is something like that, children. Let us suppose you have a small box, right? So this much box, this much big, right? This is the size of box, and both the boxes are of same size. दोनों ही box same size के हैं. Now in scenario 1, पहले scenario की अगर मैं बात करूँ, we have very thick thread, right? So this is this much of the length we take the thread, right? Now you have to keep that thread into the box. अब एक ये इतना बड़ा thread है, ये आपको इस box में fit करना है. Now the length of thread is big than the size of box, so you will not be able to keep it inside. If you want to put it straight, तो आप ऐसा नहीं कर सकते, because you are not able to fit that thread straight inside the box, right? आप उसे accommodate नहीं कर सकते. But if you coiled the same thread, लेकिन अगर इसी thread को आप coiled form में लेते हैं, right? So even it is thick, but somehow it is coiled. So if you coil it in this form, box will be able to accommodate that. तो box उसे accommodate कर लेगा. So similarly is the concept in this case of DNA. Even length of DNA is large, but DNA can be coiled, and that is how it is present inside the nucleus, right? So that in the very simple manner, it is the DNA is located inside the nucleus, right? So I hope you understand the points. Like we have discussed about the packaging of DNA helix in prokaryotic cells. We took example of E.coli, right? So they do not have well defined nucleus, and it is not like that the DNA is scattered in the cytoplasm of the prokaryotic cell. But because as the DNA is negatively charged because of the presence of negatively charged phosphate, and proteins are positively charged, so both the opposite charge attract each other, and they are accommodated or concentrated in the one region. So that region is known as nucleoid, right? So in that way, DNA in the nucleoid is organized in very large loops held by the protein, right? So this is the arrangement of packing of DNA in the prokaryotic cell. Now let us move ahead with the eukaryotic cell.